Hello, this is video number 4 for Module 6. And the topic of this module, gene linkage and genetic maps, chromosome mapping. So in the last video, I covered determining linkage between two genes based on the dihybrid test cross. And that was for genes on autosomal chromosomes. Now, we can do a similar type of analysis for genes on X chromosomes for species that have sex determined by chromosomes, for example, humans, Drosophila. So we can do the same kind of analysis for genes on the X chromosome, X-linked genes, but we have to modify the procedure a little bit. And the reason for that is females have two X chromosomes, and however, males only have one X chromosome. So that's one of the topics we'll talk about in this video. And here's a list of the 4 main topics that we'll talk about. First one is what I just mentioned, how to determine linkage and recombinant frequency for genes that are on the X chromosome. And we have to do a modified dihybrid test cross to do that. So we'll go through that and give an example of how that can be carried out. Then the next topic is this. If you know that 2 genes are linked, well, what if you know that more than 2, 5, 6, or even more are linked, right? Typical chromosomes carry hundreds of genes. Can we determine the order of genes along a chromosome given genetic cross data, dihybrid test cross, for all pairs of genes that we know are linked? In fact, we can do that, and that's called a chromosome map. And so we'll go through how to determine gene order on a chromosome based on genetic cross results. Then the third topic is, well, knowing that we can create genetic maps and that chromosomes have been sequenced, the DNA sequence has been determined, how do those 2 maps line up? Is there a good correlation between them or not? So we will review that information as well. And the final part of this video is a review of the main topics of this module. So let's start with topic number 1, determining linkage and recombinant frequency for X-linked genes. So we can do a dihybrid test cross for X-linked genes to determine linkage, and if linked, determine the recombinant frequency between those genes. And we just have to modify the cross a little bit from what we talked about for autosomal genes. So, since females carry 2 X chromosomes, the female in such a cross will have to be the double heterozygote. So here we're talking about 2 genes, A and B. They're X-linked. So if we can set up a cross with a female that's heterozygous for both, big A, big B, little b, this female has alleles in cis, both dominant on one chromosome and both recessive on the other chromosome. She will be the double heterozygote. The male, having only one X chromosome and a Y, that's what this represents, this kind of hooked chromosome. The Y doesn't have alleles for X-linked genes. So the male is going to have only one copy, and the male will serve as the tester. His X chromosome will have both recessive alleles for the genes being tested. Okay, so that's the modification we have to make. Female is always the double heterozygote, and the male is always the tester for X-linked genes. Let's look at an example. And this is an example from Drosophila, and this involved 2 X-linked genes. One is called the white gene, symbolized by W, and the 2 alleles we'll talk about is a wild-type allele that's dominant and gives red eyes. And in Drosophila, the designation for a wild-type allele is always with a with a plus superscript. The other allele is a recessive mutant allele that gives white eyes. Here, white eye phenotype here. Here's the red eye phenotype. The other gene, so the W gene is one X-linked gene. The other gene, also X-linked, is the yellow gene, symbolized by the Y. There's a dominant wild-type allele, Y superscript plus, that gives brown body. And there's a recessive null allele that gives yellow body. Here's the brown body phenotype. Here is the yellow body phenotype. Okay, so let's set up a cross, and we're going to go back to the parental generation here with a pure breeding strain that's yellow, white-eyed and yellow-bodied. Mutant allele for eye color, wild-type allele for body color. White-eyed and yellow body. White-eyed and brown body. Excuse me. White-eyed and brown body. Right? She's pure breeding. She is homozygous for both. Homozygous mutant for eye color, homozygous wild-type for body color. We're going to cross her to a pure breeding male, and the male, well, he's not homozygous. It's called hemizygous, and he's got a wild-type allele for eye color, red eyes, and a mutant allele for body color, yellow body. Okay, when you cross them, you'll get both male and female offspring, or actually, yeah, male and female offspring. This one on the left, though, the female, is what we're most interested. This female is going to get this chromosome from the mother, WY+, and this chromosome from the father, W plus Y. So this female now is doubly heterozygous, WW plus Y. She will be showing both wild-type phenotypes, red eyes and brown body color. She is a dihybrid, double heterozygote. So we can set up a dihybrid test cross with her and a tester male. This is not a tester male. This is a male from this cross. But the next slide will show the dihybrid test cross. One other point I want to make about this female is that she has alleles in trans, one mutant and one wild type, one recessive and one dominant allele on both chromosomes. Those are alleles in trans. And going back to her mother, she also has alleles in trans. So the parental arrangement here is alleles in trans. All right, let's go to the next slide, which shows the dihybrid test cross using this female and a tester male. All right, here's the female, doubly heterozygous with alleles in trans, crossed to a tester male. The tester male has only recessive alleles on his X chromosome. All right, from this cross, what are the offspring going to be? That's the F2, shown on the next slide. And what we would expect to see in a dihybrid test cross would be two non-recombinant phenotypes. And the non-recombinant should have alleles in trans, just like in the mother in this cross, and in the parent in the previous cross. Okay? Those would be the non-recombinants, or the parentals. We would also expect to see two classes of recombinants. The recombinants are going to be less common than the non-recombinants. And the recombinants will have alleles in cis, because there's been a crossover between these two chromosomes. They'll either be W, Y, both mutant alleles, or W plus, Y plus, both wild-type alleles. All right, so let's go on to the results, the F2 offspring of this dihybrid test cross. All right, here are the results. So, we see the non-recombinants, the parental types having, by the way, the results here are shown only for the F2 males, just for simplicity. We could have 4 males and 4 females, but they should be equal, so we're only representing the males on this slide. We see 1, 2, 3, 4 phenotypes of males, mutant for eye color, white eyes, brown body, red eyes, yellow body, red eyes, brown body, and white eyes, yellow body. Okay, all 4 possible phenotype combinations. The non-recombinant or parental types are showing 1 mutant and 1 wild-type phenotype, and that's because they received a chromosome with alleles in trans. That's the parental or non-recombinant arrangement. The other parental type has one mutant and one wild type phenotype. Wild type eye color, red. Mutant body color, yellow. Wild type for the eye color allele, mutant for the body color allele. Okay, so these two are both non-recombinant or parental types with alleles in trans, and we see a lot of those in the F2, for almost 4,500 of each. Okay. The other two combinations are either both wild type, red eyes and brown body, or both mutant, white eyes and yellow body. And they would be males with the X chromosome having alleles in cis. These must be recombinants because the mother for this cross had alleles in trans. So they are present in very low frequency. Whenever you see from a dihybrid test cross two phenotypes in relatively high frequency and roughly equal in frequency, those are the parental non-recombinants. And whenever you see two phenotypes very rare but also equal to each other, those are the recombinant phenotypes. So the main conclusions would be this. We definitely do not see a 1-to-1 to 1-to-1 ratio in the F2. Okay? And so that indicates that these two genes, the W and the Y gene, are linked. Okay? We can also determine a recombinant frequency from this cross. The number of recombinants, shown here in the numerator, divided by the total number of offspring, shown here in the denominator, multiplied by 100, you get a percent, and this is around slightly over 1%. Okay? The main conclusions then, these two genes, the W and Y gene, are linked on the X chromosome, and they are one map unit apart. One recombinant frequency of 1% is equal to one map unit apart. Okay, so that's how you set up a dihybrid test cross for genes on the X chromosome to test for linkage. If they depart from a 1-to-1 to 1-to-1 ratio, they are linked. And you can determine the recombinant frequency from that data. All right, now this slide is illustrating on the left side what we just went through. Starting with the parental generation, alleles in trans, generating a double heterozygote with alleles in trans, crossing it to a tester, and getting parental types 99% with alleles in trans, and recombinants 1% with alleles in cis. Okay, here's an alternate way you can set up the same dihybrid, alternate dihybrid test cross, but looking for basically the same result, the same data. If you want to test for linkage of the W and Y gene, you can start with a parental generation in which the females are pure breeding for the wild type allele. Males are mutant at both genes. You will generate an F1 double heterozygote, right? Heterozygous at both genes. However, this one has alleles in cis, unlike this one with alleles in trans. If we cross her to a tester, this is also a dihybrid test cross. We've satisfied, this should be W and Y, sorry about that. Tester would have both mutant alleles, just like over here, this tester. This dihybrid test cross then would give the same results numerically. Parental types, 99%, both of these. Recombinants, 1%. We can determine a recombinant frequency. We would conclude that these two genes are linked with a recombinant frequency of 1%. However, the recombinants here are having alleles in trans. That's because the double heterozygote had alleles in cis. And the parental types have alleles in cis, just like the double heterozygote female. Okay, so these are two different ways to set up a dihybrid test cross, one with alleles in trans, one with alleles in cis. The main results of the test cross would be the same. You would conclude these genes are linked and have a recombinant frequency of 1%. However, the details are different. and the details only differ because of the history of where these chromosomes came from in the double heterozygous female. Okay. All right, on to the second topic on the list. Chromosome mapping, determining maps of genes based on dihybrid test cross. So, we just looked at this dihybrid test cross in Drosophila with the white gene, white eye color gene, versus the yellow gene, and we came up with a recombinant frequency of 1.1, 1% recombinant frequency, and more accurately, 1.1. So the white and yellow gene have a recombinant frequency of 1.1%. If you did the same or similar dihybrid test cross with the white gene and a gene for miniature wings in Drosophila, you would come up with a much larger recombinant frequency, 32.8%, 32.8 map units. So the conclusion from this is both white and yellow are linked on the X chromosome, and white and miniature are linked on the X chromosome. That must mean that yellow and miniature are also linked to each other. Now, if they are linked, what is the order of the genes? So the next slide is showing a couple of possible orders, right? This is the same information from the previous slide, and we can determine, or we can hypothesize, two possible orders. If white and miniature are 32.8 map units apart, and white and yellow are 1.1 map units apart, the order could be this: yellow, then white, then miniature. Or the order could be this: white and yellow and miniature. That preserves these two values. How do we determine which order is the correct order? In order to do that, we have to perform a dihybrid test cross using the yellow and miniature genes. If we get a recombinant frequency greater than 32.8, the frequency between white and miniature, then this is the order. It means that yellow is away and the white gene is in the middle. If we get a recombinant frequency less than 32.8, that means that the yellow gene is in the middle. Okay? So this is how you can determine orders of multiple genes along a chromosome by setting up pairwise dihybrid test cross between all of the genes that are involved. If we did this dihybrid test cross between the yellow and the miniature gene, we would come up with this order. Here are the recombinant frequency values: yellow to white, 1.1, white to miniature, 33.8. If we do yellow to miniature, we get a value greater than 33.8. That means that this is the order along the chromosome. We can do that for a number of other genes as well. So they're shown here: the R gene, the V gene. If we do all pairwise combinations of genes in dihybrid test cross, we can come up with recombinant frequency values for each of them. And then based on these recombinant frequency values, we can determine the order of all 5 of those genes along the chromosome. So this is how you determine a genetic map. And I have a problem in the problem set which challenges you to determine the map of a chromosome based on this recombinant frequency data. So I will be creating a video of that problem as well, and I will be posting that on Monday, April 6th. So I'll be looking for that tomorrow. Okay. Now, so the recombinant frequencies of different pairwise combinations of genes that are linked, excuse me, let me go back here, can be determined to generate a chromosome map. Now, the way that that's possible is if the recombinant frequency is proportional to the distance apart on the chromosome, right? A high recombinant frequency indicates that genes are far apart on the chromosome. A low recombinant frequency indicates that genes are close together. And we're assuming that far apart means there's more opportunity for crossover to occur. So that would produce a higher recombinant frequency. And closer together means a lesser opportunity for crossover to occur. So short map distance means few recombinants. Long map distance means a lot of recombinants. So the assumptions that we're using in creating genetic maps are that recombination, crossing over, occurs randomly along the chromosome. That there are no places where there's a very high frequency, unlike other places on the chromosome. It's random and can occur with equal frequency at any point. The other thing that we're assuming is that the recombinant frequency values are additive, and that this map, in order to create orders, is going to rely on this additivity. So the additivity point is not exactly true, right? If it were true, then these map distances, yellow to white, 1.1 map units, white to miniature, 33.8 map units, would sum up to 34.9, but they don't. So usually there's some degree of error. There's not exact additivity. However, the important thing is this value is greater than this value. And so that must mean that the yellow gene is over here and not between white and miniature.